Once again, greetings to all my viewers from Dr. Raktim Mukherjee, Assistant Professor and Head of the Department, Shri P.M. Patel Institute of PG Studies and Research in Science, affiliated to Sardar Patel University. In continuation to our earlier videos on epinephrine signaling pathway and a related talk on how the epinephrine signaling pathway is switched off, we still continue our journey with GPCRs, that is G protein coupled receptors, and in this video we shall discuss the G protein coupled receptors, the phosphoinositide cascade, and this cascade uses two different second messenger system that of course will feature our succeeding videos. So the title of the talk is Phosphoinositide signaling pathway. The snapshot summary: this pathway also involves G protein or 7-transmembrane receptor, abbreviated as 7TM receptors. One of the example in this pathway, earlier example we talked about epinephrine in epinephrine signaling pathway. Here we would talk about angiotensin 2 receptors. The angiotensin 2 receptors in our body binds a primary messenger which is used in the pathway. To just give you a background, angiotensin 2 is a peptide hormone that controls our blood pressure. Students, our kidney secretes a hormone called as renin. This hormone renin from the kidney converts inactive angiotensinogen to active angiotensin 1, and the enzyme is angiotensin converting enzyme, and angiotensin converting enzyme in turn converts angiotensin 1 to angiotensin 2, and this angiotensin 2 is responsible for maintenance or control of blood pressure, and that is why this signaling pathway is very important, because it maintains the blood pressure of our body. Now, intracellular messengers of phosphoinositide cascade. Like cyclic AMP cascade, this pathway also converts extracellular signals into intracellular ones. The intracellular messengers formed by activation of this pathway arise from the cleavage of, this is important. First we will go to the figure, and it leads to cleavage of PIP2. This is phosphatidyl inositol 4,5 bisphosphate. I have already highlighted phosphatidyl inositol 4,5 bisphosphate, and this molecule PIP2, PIP2 is a membrane bound molecule, is cleaved into two subsequent parts. One of course is inositol triphosphate and diacylglycerol. So this is your diacylglycerol, the bottom one, and inositol 1,5,1,4,5 triphosphate, that is IP3. Remember students, whereas diacylglycerol is bound to the membrane, IP3 will translocate towards the cytosol. Now, when you have a signaling through a distinct G protein, let's important signaling through G protein, the receptor, you have a beta adrenergic receptor which activates G protein G alpha stimulatory. This is something we already covered. Beta adrenergic receptor activates G protein, that is G alpha stimulatory. Angiotensin 2 receptor activates a G protein called as alpha Q. So this is the difference, that beta adrenergic receptor, it activates G protein G alpha stimulatory, and angiotensin 2 receptor, that is the current pathway that we are studying, here activates the G protein G alpha Q. So the G proteins involved in both pathways, the phosphoinositide pathway that we are studying now, is distinctly different from the earlier video where we talked about epinephrine signaling pathway. So let's go to the very first step of phosphoinositide pathway. The first step is cleavage of PIP2. So this is the diagram. We have the membrane bound enzyme phospholipase C beta. This is phospholipase C gamma, 2 fragments. So try to understand that in the GTP form, the G alpha Q, when it is bound with GTP, it activates the beta isoform of the enzyme phospholipase C. This is the beta isoform of the enzyme phospholipase C. When the beta isoform of phospholipase C gets activated, it gets activated when the GTP is bound to G alpha Q. Only then it gets activated. Imagine it gets activated because it is bound with G alpha Q, that is GTP. So we take our sojourn into the next few steps of the pathway. The next few steps of the pathway, we have the cleavage of PIP2. Phospholipase C cleaves the membrane lipid phosphatidyl inositol 4,5 bisphosphate into 2 messengers. You can see from the diagram that PIP2 is cleaved into 2 messengers. One will remain in the bound with the membrane, that is diacylglycerol. Another will translocate to the nucleus, that is inositol triphosphate. So phospholipase C cleaves membrane lipid phosphatidyl inositol 4,5 bisphosphate into 2 messengers. The first one is diacylglycerol. This is your diacylglycerol, and the next one is inositol 1,4,5 triphosphate. This is your inositol 1,4,5 triphosphate. You can see in the parent molecule, that is phosphatidyl 4,5 bisphosphate PIP2, you had both the inositol triphosphate portion and the diacylglycerol portion. So this bond is broken. There is a cleavage of this molecule. Once this molecule is cleaved, which molecule? The PIP2. Once this molecule is cleaved, only then it will lead to the conversion of DAG into IP3. Remember students, diacylglycerol will remain in the membrane. You can see diacylglycerol is bound to the membrane, whereas IP3 or inositol 1,4,5 triphosphate will diffuse away from the membrane. It will remove towards the cytosol. Now we study the role of inositol 1,4,5 triphosphate. What is the role? It is soluble and it diffuses away from the membrane. You can see it is soluble and it diffuses away from the membrane. It is no longer membrane bound. This diffusion away from the membrane causes a rapid release of calcium from the intracellular stores in the endoplasmic reticulum. This I will talk in my next video, that is calcium signaling pathway video, that why calcium remains in the endoplasmic reticulum of any other cells and sarcoplasmic reticulum of the muscle. So it causes a rapid release of calcium from the intracellular stores of the endoplasmic reticulum. This is your endoplasmic reticulum. You have huge stores of calcium, and those calcium is released from endoplasmic reticulum whenever IP3 binds to the calcium channels, the membrane bound calcium channels, endoplasmic reticular calcium channels. You have a specific IP3 gated calcium channels present in the endoplasmic reticulum, and IP3 specifically, this IP3 specifically binds to those IP3 gated calcium channels. Once IP3 binds, the endoplasmic reticulum calcium channel opens, and calcium which is stored in the endoplasmic reticulum will move from endoplasmic reticulum to the cytosol. So to summarize, inositol 1,4,5 triphosphate helps in mobilization of calcium from endoplasmic reticulum to the cytosol. Once the level of cytoplasmic calcium elevates, what it does, it triggers various physiological events such as smooth muscle contraction, the breakdown of glycogen to glucose, and release of vesicle, which is a very important prominent pathway in neurotransmitters release or synaptic transmission. Remember students, even the calcium that is extruded out from the endoplasmic reticulum, that calcium also acts as a signaling molecule, which you will see in the next video. So calcium is also a signaling molecule. Why it is a signaling molecule? Calcium can bind to proteins called as calmadulin and enzyme such as protein kinase C. So calcium can bind to both calmadulin as well as protein kinase C. So that completed, we talked about PIP2, the membrane bound PIP2. It breaks down into two moieties. First moiety was inositol triphosphate. Inositol triphosphate we have discussed. Now we will discuss the another breakdown product, that is diacylglycerol. So diacylglycerol, this remains in the plasma membrane. Diacylglycerol remains in the plasma membrane. See, it is bound to the plasma membrane. And by remaining in the plasma membrane, diacylglycerol activates protein kinase C, also called as PKC, the protein kinase C. Once protein kinase C is activated, you can see diacylglycerol activates this protein kinase C. Once protein kinase C is activated, protein kinase C will phosphorylate serine and threonine residues in many target proteins, and the specialized diacylglycerol binding domains of kinase will require calcium, and that calcium again you are getting through inositol triphosphate pathway. So inositol triphosphate increases the calcium concentration, and the calcium facilitates the diacylglycerol mediated activation of protein kinase C. Thus both inositol triphosphate and diacylglycerol act transiently. It should be diacyl, DAG it should be. It acts transiently. This is DAG. This DAG act transiently, that is for a brief period of time. Why they act transiently? Because they are converted into other species of phosphorylation or other processes, and that is the reason the action is very slow. Now try to understand. Any guess who this fast bowler is? Anyone? This is the legendary West Indian fast bowler Malcolm Marshall. On the right side, another West Indian fast bowler, Michael Holding. Whenever a fast bowler bowls, the fast bowlers work in tandem, that is to harass the batsman in a maximum possible way. They work in tandem, that is one fast bowler always changes the ends and another fast bowler joins, that is they bowl in tandem. Similarly, the diacylglycerol or inositol triphosphate also work in tandem, that is, they work together. What they do when they work in tandem? First, inositol triphosphate increases the level of calcium, and once the level of calcium is increased by inositol triphosphate, this calcium which was increased by inositol triphosphate, that calcium will facilitate the diacylglycerol mediated activation of protein kinase C. So we say they both work in tandem. IP3 increases calcium, and that increased calcium will facilitate the diacylglycerol mediated activation of protein kinase C. So students, once again to summarize, you have PIP2. This PIP2 is cleaved to two molecules, IP3 and diacylglycerol. Diacylglycerol is membrane bound. Inositol triphosphate translocates to the cytosol. Inositol triphosphate binds to the calcium channels present in the endoplasmic reticulum. Endoplasmic reticulum has huge stores of calcium, and by binding to the endoplasmic reticulum calcium channel, it mobilizes calcium from ER to the cytosol. This calcium in the cytosol will facilitate the diacylglycerol mediated activation of protein kinase C by working in tandem. Together, this carries out the various signals transducing process. Even the calcium from endoplasmic reticulum, the calcium alone can also act as a second messenger carrying out signal transduction pathway. Thus, we complete the inositide phosphate signal transduction pathway. Thank you very much for your attention. Our next video shall feature on the calcium signaling pathway